Microbial nutrition. In this lecture, we'll be looking at what microbiology is, what microorganisms are, and how we can grow microorganisms. Microbiology is simply the study of organisms that are too small to be seen clearly with the naked eye, in which case they are being observed under a microscope. Whereas microorganisms are the organisms that are studied in microbiology. So these organisms include bacteria, viruses, fungi, protozoa, and the likes. In this particular lecture, we will be more interested in factors that influence growth. When we say growth, we mean acquisition of biomass that leads to cell division or reproduction, and not merely by an increase in size. And so if we say a microorganism has grown, then we are saying they have multiplied in numbers, maybe from 1 to 2 to 4 to 16 to 32 to 64, and the list continues. To be able to link, this is very important because to be able to link a particular disease to a particular microorganism, for instance, Salmonella typhi, responsible for causing typhoid fever, then we should be able to grow Salmonella typhi on a growth media. That is why we need to know the factors that would support the growth of these microorganisms in an environment outside its origin. And so there are some surfaces that we need to be aware as we go through this lecture. We have a file and a tolerant. Anytime we say a file, then we are describing a condition that will permit growth. Whereas when we say tolerant, we are describing a condition that an organism can survive, but they cannot necessarily grow. So an example of a file that has been used, as I explained early on, is a thermophilic bacteria that would grow under elevated conditions, whilst a thermotolerant bacteria will survive at that elevated temperature condition, but they would need to grow at a lower temperature. Let's look at factors that influence microbial growth, looking at obligate and facultative. The word obligate simply means strict, whilst facultative simply means they are flexible. So means they are flexible. An obligate organism means that a given condition is required strictly for that microorganism to grow. Whereas in facultative, it means that the organism can grow, but under that condition only, but they do not require it for survival. So the term facultative is often applied to suboptimal conditions. Suboptimal means that the condition required for that organism to grow is not reached, but it can still survive. For example, an obligate thermophilic requires elevated temperature for growth, whereas a facultative thermophile may grow in either elevated temperature or even a much lower temperature. Temperature is a very important growth condition in microbial growth studies. So when you go to any microbiology lab, there is what we call an incubator. What the incubator does is that the incubator is set to make sure that a particular temperature set remains for a very long time so that microorganism can grow. So a microorganism that can grow only at a temperature of 20 degrees Celsius, that incubator has to be set for that temperature for the length of time, being it 24 hours or 48 hours that microorganism need to grow. So we say most bacteria will grow throughout a range of approximately 20 degrees Celsius, with the maximum growth rate occurring when that temperature is optimal. So what we are saying is that there could be various growth temperatures that they could grow ranging around 20 degrees Celsius, but for the organism to grow best, that temperature must be obtained. Psychrophiles. Psychrophiles are microorganisms that grow at temperatures of 0 degrees Celsius, although their optimal temperatures could be between 0 to minus 15 degrees Celsius. We have psychrotrophs. Psychrotrophs can also grow at temperatures between 0 to 10 degrees Celsius, but then their optimum growth temperatures range between 20 to 30 degrees Celsius, while their maximum temperatures is at 35 degrees Celsius. Mesophiles. Mesophiles also would grow at optimum temperature around 20 to 45 degrees Celsius. Then we have the moderate thermophiles that grow between 55 to 65 degrees Celsius, and then the extreme thermophiles that are hyperthermophiles that needs elevated temperatures of between 80 to 113 degrees Celsius. pH. pH simply means the level of acidity or an alkalinity of a particular environment. Microorganisms also need specific pH within which they can grow optimally. So, based on pH categories or classification, microorganisms have been divided into 3 main sections. We have the acidophiles, neutrophiles, and alkalophiles. Acidophiles will grow optimally between temperatures of between pH of 0 and 5.5. Neutrophiles will grow optimally between pH of 5.5 to 8, whereas alkalophiles will also grow optimally between pH of 8 and 11.5. But like I said, these pH are so critical if we need the microorganisms to grow optimally in a given media. Let's look at salt concentration. Salt concentration is simply the amount of, as it were, sodium chloride related properties that you find in a given solution. Microorganisms, like I said earlier on, will also require a particular salt concentration to be able to grow, depending on which group that we're looking at. If they are halophiles, then it means they love salt and they will require elevated salt concentrations to grow. Often 0.2 molar ionic strength or even greater may be conducive for them to grow. Example of these halophiles may include the halobacterium. Then we have the osmotolerant or halotolerant. These are a group of microorganisms that grow well over a wide range of salt concentration or ionic strength. For example, Staphylococcus aureus will need those temperature ranges to be able to grow. Oxygen. Oxygen is a very important requirement for living organisms, and that includes microorganisms. But for some of them, the presence of oxygen would kill them. So those that love oxygen are known as aerobes, and those that hate or that will be killed by oxygen are called anaerobes. However, I must be quick to state that the demand for oxygen varies between microorganisms, whether they are straight aerobes or they are straight anaerobes. Now, for straight aerobes, they require oxygen of about 20% in terms of concentration to grow, whereas straight anaerobes or anaerobes, they will grow only in the absence of oxygen, and they cannot grow in the presence of oxygen. So it means for those organisms, if you expose them to oxygen, they are killed. We also have the facultative anaerobes. These facultative anaerobes will grow best in the presence of oxygen, but they are also able to grow at reduced rate or in the absence of oxygen. Aerotolerant anaerobes. We have some anaerobes that are aerotolerant. What they do is that they can grow equally well in the presence or the absence of oxygen, which means they can tolerate the presence and absence of oxygen. And then we have the aerophiles. The aerophiles will require reduced concentration of oxygen between 2 to 10%, for growth. Nutrient requirement. Apart from those environmental conditions, nutrient requirements are also critical for microorganisms to grow. Let's look at carbon source as a nutrient requirement. There are some microorganisms that are within the range of autotroph, and they can use carbon dioxide as a sole carbon source. They do that through carbon fixation when green plants are able to fix carbon dioxide directly from the atmosphere in the presence of sunlight and water through photosynthesis so that they can capture this carbon into the plant, and then these microorganisms can feed or use them for their source of growth. Heterotrophs. Heterotrophs are microorganisms that require an organic source, but they can also use carbon dioxide as a carbon source for their nutritional requirement. Nitrogen source. There are some microorganisms that also require nitrogen source, but in other form. There are some that require organic nitrogen primarily from catabolism of amino acids, or even oxidized forms of inorganic nitrogen, primarily in the form of nitrates and nitrites. There are some that also require nitrogen source in the form of reduced inorganic nitrogen in the form of ammonia. And there are some that also require this nitrogen source when they are dissolved as dissolved nitrogen through nitrogen fixation. Phosphate source. There are some microorganisms that also require phosphate either in organic form or inorganic form. Sulfur source. There are some microorganisms that can also use sulfur as nutritional requirement either in the form of oxidized inorganic sulfur, reduced inorganic sulfur, or even elemental sulfur. Special requirement. There are some group of microorganisms that will also require special growth nutrients in the form of either amino acids, nucleotide base, or even enzymatic cofactors or vitamins. Microbiological media. A microbiological media is any growth substances that contains the right proportion of nutrients and growth conditions that will support the growth of a particular microorganisms. And so we say we have different microbial media for different group of microorganisms. This broad group of microbial media has been divided into two main portions. We have those that are chemical defined and those that are also complex media. Chemical defined media. To be chemical defined media, it means your exact chemical composition must be known. Example is the minimal media that are used in bacterial genetic experiments. Then we have the second type, that is the complex media. The complex media is also a media where the exact chemical composition is, however, not known. They often would consist of a plant or animal extract such as soybean meal, milk protein, or any form of substances that are needed. They often include most routine laboratory media such as tryptic soy broth, and the list continues. That is not all. We also have selective media and differential media. A selective media is a type of media that contains growing agent that will inhibit, or that contains a growth agent that will inhibit the growth of certain bacteria whilst permitting the growth of others. They are frequently used to isolate specific organisms from a large population of contaminants. For example, if we took a human sample from a patient suffering, say, cholera, and we are interested in growing Vibrio cholerae from that particular specimen or sample that we are taking, we will go in for a media that will support only the growth of Vibrio cholerae but suppress all other microorganisms.